Okay, good evening, everybody. And a warm welcome to you all. Welcome to UEA. It's great to see you here today on a Tuesday evening in November. My name is Laura Bowater, and I'm Professor of Microbiology Education and Engagement at UEA's Norwich Medical School. So I feel quite at home here introducing Matt and his lecture. And it's especially timely, I think, that we're all here today to listen to Matt's inaugural, because this week is World Antibiotic Awareness Week. So I just wanted to say my congratulations to UEA on your immaculate organisation and timing of this particular event to clash with that. It's my great pleasure to be introducing Matt Hutchings today. Matt developed his interest in biology while still at high school, and he gives credit for this to a combination of great teachers, obviously, and a book called DNA for Beginners. Has everybody got space? Great. Hello. So, after leaving school at 18, Matt worked in a hospital lab at St Heliers Hospital in London, where a doctor encouraged him to go to university to have some fun. In the words of Matt, and this I think shows Matt's age, he says, "I wish I could remember his name, because it was the best advice that I ever had. I grew up on a council estate and my parents didn't go to university, so I had no idea what I was getting myself into when I moved to Portsmouth to study biology, but I loved everything about university life. It was a life-changing experience. And when I graduated, I decided to move to Southampton to do a PhD in molecular biology, and then in 1998, I moved to Norwich for a postdoc at UEA. Now, Matt went on to do a second postdoc at the John Innes Centre in Norwich, and that's actually where we met. So Matt started working in Mark Butner's lab, changing fields to work with streptomyces. And streptomyces are amazing microbes. They make natural products that provide us with most of the antibiotics that we use in human medicine. 4 years later, Matt decided to go it alone, and he applied for two possible positions, a lectureship in the University of Strathclyde in Glasgow, and a Research Council's UK Fellowship at UEA. He was offered both, and after much agonising, chose UEA, which I have to say, you obviously made the right decision.
I think so. And
because he wanted to carry on with streptomyces research, he said in his words, "Norwich is the best place in the world to do it," and I have to agree with you about that. So it was at UEA that Matt met fellow researcher Doug Yu, and I see Doug's in the audience, and he learned about leafcutter ants, and he discovered about their use of antibiotics that are made by symbiotic bacteria that these ants carry on them. Doug and Matt successfully obtained a UEA-funded PhD studentship, and they set about trying to identify the antibiotics produced by the ants and used by the ants. So 10 years later, they're still working on the use of natural product antibiotics, and they're hoping to identify new molecules which might be developed as drugs. Now, antibiotics have been used and misused over the past 100 years. And tonight, Matt will talk about his research and his quest to discover why bacteria make antibiotics and why have insects and plants been able to make use of these antibiotics without experiencing the same problems as we have, which is drug resistance. So could symbiotic bacteria offer us new prospects for drug discovery in the future? I think we're going to find out tonight. So please join me now in welcoming Professor Matt Hutchings to give his inaugural lecture here tonight.
Hey, thank you, Laura, for the kind introduction, and thank you all for coming. It's great to see so many people here. I think this is the biggest audience I've ever had for a talk. So I have got a bit of a throat infection, which is kind of ironic. I think it's viral, not bacterial, so I won't be taking antibiotics. So before I start, I'd like to thank or acknowledge my research group. So this is my current group, a photo we took yesterday on the beautiful UEA campus. So this is a fantastic bunch of PhD students, 7 PhD students and 2 postdocs who did a lot of the work that I'm going to talk about today. So not only are they brilliant experimentalists, but every time I ask them to go out and do public engagement and go to schools and talk to kids about science, they always jump at the chance. So they really are great to work with. And I would say to all of them and people in the audience that if a council estate kid like me can go to university and become a professor, then all of you can. So don't let anybody tell you you can't. Okay, so I'm going to talk about antibiotics, the history of antibiotics, and then I'm going to talk about some of the work we've been doing to try and understand how they're used in nature, and some other work we've done to try and discover new antibiotics that could be used in medicine. And these little beasts that you might have seen outside are going to feature quite heavily in this story. Okay, so antibiotics are something I think we all take for granted. Is there anybody in the room that's never taken antibiotics? Put your hand up. Everybody in the room has taken antibiotics, which is amazing. So sometimes I do this with undergrads, and there are people that have never taken antibiotics. But it's quite rare. We can get them very easily. You probably only need to take them if you have a life-threatening infection, but people take them to cure sore throats and minor ailments. Actually, modern medicine would be impossible without antibiotics. Things like chemotherapy, transplant surgery, the risk of infection would be so great that it probably wouldn't be possible, wouldn't be worth the risk. They're amazing because they're selectively toxic. So they kill living microbes inside of your body without killing you, without harming you, which is quite amazing. They cure disease. There aren't many drugs out there that actually cure disease, and sometimes they can do it in a week, less than a week. Most drugs that you take manage conditions, so cancer, therapeutics, things like that, and you take them for the rest of your life. It's estimated they've increased average life expectancy by 23 years, which is half of my lifetime. So that's kind of a sobering thought. And as Laura said at the start, about 70% of the antibiotics that are used in the clinic are natural products. And this means they're naturally made by microbes. We call them secondary metabolites. That's the kind of scientific name. They're made by bacteria or fungi, and they make them to kill other bacteria and fungi in the soil. So the soil is a really harsh place to live. There's not a lot of food about. So anything that gives these microbes an advantage in the soil to get more food is a good thing. So antibiotics are probably chemical weapons. And these secondary metabolites have activities sometimes against bacteria, fungi, viruses, worms, insects, plants, everything that lives in soil, these bacteria can kill. Okay, so the history of antibiotics really starts with this guy, a German chemist called Paul Ehrlich, who just over 100 years ago discovered the first antibiotic. And this was a synthetic antibiotic, so it was man-made. It's called salvarsan. It was based on arsenic, so an organic salt of arsenic. And Paul Ehrlich came up with the idea of chemotherapy or selective toxicity, the idea that you could maybe develop molecules that could kill infectious bacteria and cure disease without killing the patient. So he did this very successfully. At the time, this was the most widely prescribed drug on earth. It was used to cure syphilis, which sent you crazy, sent you mad, and eventually killed you. So this was quite a step forwards. Salvarsan was superseded and made redundant by the discovery of penicillin by Alexander Fleming in 1928. And Alexander Fleming famously was growing Staphylococcus aureus bacteria on this petri dish on an agar plate. And he noticed that a mold started growing on this agar plate. Okay, so these are the Staph aureus bacteria at the bottom, and this is a penicillium mold, a contamination. And what he noticed, Fleming's genius really was to notice that the mold has a zone of inhibition around it where the bacteria can't grow. And he realized that that mold was secreting a compound, producing a compound that had antibiotic activity. It was killing bacteria. And he called it penicillin. And then two chemists called Howard Florey and Ernst Chain figured out how to grow the penicillium mold in huge cultures and purify that antibiotic to make it very, very pure so it could be turned into a drug. And penicillin and beta-lactam antibiotics like penicillin are still widely used today. I think it's interesting to reflect on the fact that thousands of years ago in ancient Egypt and ancient Greece, healers, traditional healers, were hanging up bread to go moldy, and they would rub that moldy bread on wounds, people that were wounded, to prevent infection. And they were probably using penicillin, but obviously they didn't know what the molecule was. So antibiotics have actually been used for millennia, but only recently, only in the last 100 years, have we used them in very purified forms. So next up is Selman Waxman. So Selman Waxman was a Ukrainian scientist working in the US, the United States. And he heard about Fleming's discovery of penicillin, and he thought, well, maybe the bacteria that I work on, Streptomyces bacteria, that are widespread in the soil, so they're in every soil on Earth, they give the soil its smell, maybe Streptomyces make antibiotics as well. So he started looking at these Streptomyces bacteria to see if they had antibiotic activity, and he discovered lots of new antibiotics, including streptomycin. Streptomycin won him the Nobel Prize for Medicine in 1952, and streptomycin was remarkable because it was the first treatment for tuberculosis. So TB is a disease that has plagued humans for millennia, probably tens of thousands of years. So Egyptian mummies have evidence of tuberculosis. So this was a killer up until the discovery of streptomycin. And it's surprising that streptomycin is still used to treat tuberculosis today, which kind of tells you probably how few antibiotics we really have. Okay, there aren't many antibiotics that can kill tuberculosis. Again, in folk medicine, so in Russia, peasants were warming up soil to use as a poultice to put on wounds for hundreds, possibly thousands of years. So again, soil is full of antibiotic-producing microbes, and warming the soil up protected people against infection. So again, antibiotics have been used for a long time. Okay, so secondary metabolites, these natural products as antibiotics. So they make up about 70% of antibiotics, as I said at the start. Streptomyces and their close relatives, bacteria that we call actinomycetes, make about 60% of those antibiotics. Fungi make around 30%, including penicillin, and all other bacteria together make less than 10%. So these really are, Streptomyces really are remarkable bacteria. We call them antibiotic factories because they make so many different antibiotics. Most bacteria are single cells, so E. coli and Staph aureus, Staphylococcus aureus, they grow as single cells and they divide when they reproduce. Streptomyces are quite weird bacteria because they grow very much like a fungus. They grow as these long hyphal filaments. And Streptomyces, the Latin name means twisted fungus. Okay, so when they were first discovered, people thought that they were fungi. They're actually about 1,000 times smaller than they're bacteria. So Norwich has a very proud history of Streptomyces research, as Laura alluded to. So you probably know, I hope you know, that we're part of the Norwich Research Park. So the university, 3 research institutes funded by the government, the Earlham Institute, the John Innes Centre and the Quadram Institute, the Teaching Hospital, and the Sainsbury Laboratory, which is part of UEA. So 50 years ago, Sir David Hopwood, who I'm pleased to see in the audience, moved from Glasgow to Norwich. And at this time, so the university was open 55 years ago, and it started teaching biology about 50 years ago, I believe. And John Innes, Sir David Hopwood was appointed as the John Innes Professor of Genetics at UEA. He established a lab at the John Innes Centre, and he taught on the biology degree at UEA. And David is really credited with being the founding father of our field of Streptomyces genetics and Streptomyces biology. So he was joined shortly after by Keith Chater, who also did a lot of the seminal early work on Streptomyces biology, and by Mervyn Bibb, who's also in the audience. So Merv, I believe, was one of the first undergraduates on the biology degree at UEA, and was taught by David, and then went to California, I think, after his PhD. He did a PhD with David, went to California, came back and started a lab at the John Innes Centre. And Merv really developed lots of the genetic tools that we use in Streptomyces and did a lot of amazing work on the regulation of antibiotic production. Mark Butner did a postdoc with Mervyn Bibb and still runs a lab at the John Innes Centre. And I'll get to the point. I did a postdoc with Mark Butner, so there's a direct line, I think, from David all the way through to me. And I like to scare Mark. I don't know if he's in the audience, but I like to call him my science dad, which really freaks him out. So there's still 6 Streptomyces groups on the Norwich Research Park, 4 at the John Innes Centre and 2 at UEA. Susan, myself, and Gabriella Kellerman were all postdocs with Mark, so we all trained with Mark. And in fact, a lot of the people that work on Streptomyces bacteria around the world came through the John Innes Centre at some stage. They trained here in Norwich. So Norwich is very famous in the field of Streptomyces biology. Barry Wilkinson and Andy Truman came from Cambridge, so they're not part of the Norwich crew. They're kind of outsiders, but they've joined us now. They've seen the light and they've moved to Norwich. And Barry Wilkinson moved here about 5 years ago, and we collaborate quite closely. So Barry is a chemist, so a lot of the chemistry I'm going to talk about later on was done by Barry Wilkinson's lab, so in collaboration. Just going to have a drink. Okay, so this slide shows a timeline of antibiotic discovery. And we talk about a golden age of antibiotic discovery, which is the 1940s and 1950s. So antibiotic discovery really peaked in the mid-1950s. So I told you at the start about salvarsan. So this is the arsenic-based compound that was used to treat syphilis, superseded by the introduction of penicillin. So the first two groups of antibiotics to be discovered were synthetic, man-made. And the sulfur drugs, which are still used today, the sulfonamides, are interesting. So the first version, Prontazil, was discovered by another German chemist working at Bayer in the 1930s, Gerhard Domach, and he won a Nobel Prize for medicine for discovering for developing Prontazil, and actually used it to save his daughter's arm from amputation. So before the antibiotic had been approved for clinical use, he treated his daughter with it because she had an incurable infection and she was going to lose her arm, and he actually saved her arm from amputation. The golden age really was kick-started by the discovery of penicillin, and people realized that microbes make antibiotics. So bacteria and fungi living in the soil make all these amazing molecules that we could potentially use as antibiotics. So everything in red is a natural product that comes from bacteria or fungi. By the 1960s, the discovery efforts had started to decline. And the problem was that when you first look in soil, so people looked in soils all over the world, they would go out to weird and wonderful places, bring back soil when they went on holiday, discover new strains of bacteria or fungi making new antibiotics. And when you first look in soil, you discover new antibiotics. But the more you look in the same place, the more you rediscover the same molecules. And scientists started to rediscover the same antibiotics over and over again from the 1960s onwards. And they assumed, because of this problem of rediscovery, that they'd found everything there was to find. So they assumed that all the antibiotics had been found, all the natural product antibiotics had been found. And the big drug companies switched to trying to make synthetic antibiotics. So they spent decades and billions of dollars trying to develop man-made antibiotics like the sulfur drugs without very much success. Okay? And now we know from genome sequencing, which I'm going to talk about, that actually we've barely touched the surface. There are loads of antibiotics out there waiting to be discovered. And interest is growing again in natural products. So this shows a timeline of antibiotic resistance or antimicrobial resistance. And this is where bacteria become resistant to the antibiotics we use in the clinic. So under selective pressure, they evolve very quickly to become resistant, and then we can't treat them with the drugs anymore. So Fleming warned in his Nobel Prize speech in 1945 that if we misuse antibiotics like penicillin, bacteria would rapidly become resistant and they wouldn't work anymore. And he was absolutely right, but we went on to misuse antibiotics anyway. So about 50% of them have been used in agriculture to promote the growth of cattle and other livestock because they gain muscle mass much quicker if you put antibiotics into animal feed. So not even used to treat disease. And there's been a lot of misprescribed antibiotics over the years, I guess. It used to be if you went to the GP with any kind of infection, you could get an antibiotic. And I'm pleased to see now it's very difficult to get antibiotics prescribed by GPs. So pretty much, within 10 or 20 years of antibiotics being introduced, bacteria become resistant. And of course, now we have things like multi-drug resistant tuberculosis, colistin-resistant E. coli. They're very, very difficult to treat because there aren't really any antibiotics left to treat them. And one of the problems, really, with antibiotic discovery is financial, it's economic. So it's very expensive to try and develop a new antibiotic and put it through clinical trials. So GlaxoSmithKline, people will tell you it costs $1 billion to get an antibiotic from the discovery stage through the clinic to the market. And the government were quite savvy. They appointed an economist, Lord Jim O'Neill, to lead a report or publish a report into antimicrobial resistance a few years ago. And the O'Neill report said that if we don't act now, then by 2050, 10 million people a year will die from infectious diseases. So we'll basically go back to the stage we were at 150 years ago before we had any antibiotics. There'll be no drugs left to treat people. And not only will the cost in human lives be enormous, so a bigger killer than cancer, but obviously the cost to healthcare systems will be crippling. So it's really important that we start to develop new antibiotics now. And the question is, who's going to pay for it? Because nobody really wants to fund it. Okay, so there's a lot of good news in this story. That's kind of the bad news. But we have a lot of reasons to be optimistic. And one of the reasons was this paper that came out in 2002. So I'd been a postdoc at the John Innes Centre for about 6 months, I think, when this came out. And this was the complete genome sequence of Streptomyces coelocolor. So this was the first Streptomyces genome sequence. It was led by David Hopwood and Keith Chater with colleagues at the Sanger Centre in Cambridge. And the complete genome sequence, basically you sequence all the DNA in this bacterial strain, and the DNA is the instructions for life. Okay, so the DNA, if you can read the DNA, you can understand all the molecules that these bacteria can make. And the striking thing, which I highlighted here, is the 7,825 predicted genes include more than 20 clusters coding for known or predicted secondary metabolites. So at the time, it was known that Streptomyces coelocolor made 4 or 5 secondary metabolites. But it turns out it's got the instructions to make many more. They're just never made under lab conditions. So in the wild, when they're living in the soil, they must make all of these secondary metabolites to kill other organisms in the soil. But when we grow them in the nice warm conditions in the lab, they don't make them. And the idea here is these silent or cryptic secondary metabolites, these antibiotics that are never made, if we could switch them on, we could discover lots more antibiotics. And there are about 500 or 600 known species of Streptomyces, thousands of strains, and all of them probably only make 25% or less of their secondary metabolites. So there's loads of potential for antibiotic discovery. So this was a pretty seminal moment in my time at the John Innes Centre. And the other seminal moment was I got married. So this was actually more exciting than the Streptomyces genome sequence. My wife's in the audience. So we got married in 2002. I have very brown hair in this photo. We actually got married, bought a house, and I turned 30 all in the same week. So that was a pretty intense week. And actually today I'm giving this inaugural lecture, and tomorrow we're moving house. So these things seem to come in all at the same time. Okay, so the future of antibiotic discovery is genome mining. And this means sequencing the genomes of these bacteria, looking at the instructions to see what kind of secondary metabolites they can make, and then trying to switch them on so that we can discover new antibiotics. So this is a strain that we work with, a new species of Streptomyces that we called Streptomyces formicae. I'll talk about it later on. We isolated it from an ant, so formicae means ant. Each one of these colored dots basically represents a cluster of genes encoding a secondary metabolite, potentially an antibiotic. So this strain is remarkable. It encodes at least 46 different secondary metabolites. It only makes 2 of them in the lab, so 44 are never switched on. The 2 that it does make are very potent antibiotics that I'll talk about a little bit later on. So obviously, if we could switch on everything in this strain, then we could discover loads of cool new molecules. And the other thing is to look in new environments. So in the 40s and 50s, most people looked in soil. Obviously, the planet is covered, 2/3 of the planet is covered with oceans, and now we have the ability to actually sample sediments from deep under the sea, thousands of meters under the sea, which have lots of interesting microbes growing down there and making lots of interesting molecules. Hyper-arid deserts, so I know Merv has done a lot of work on the Atacama Desert in Chile, which is one of the driest places on Earth, and I'm going to talk about these little beasts, these leafcutter ants, and how they use antibiotics and how we might discover new molecules from them. So here, again, Laura alluded to this in her introduction. So Doug is in the audience. Oh, he's over there. So Doug is in the audience. So when I moved to UEA, after 4 years at the John Innes Centre, I moved to UEA in 2006, January 2006, to take up a Research Council's fellowship in the School of Biology. And one of the first people I met and got talking to was Doug Yu. And Doug and I got talking at a school meeting. So apologies to John Tenner, because I think you were head of school then. But actually, we were really bored in this school meeting. And we started talking about Apple computers and about music. And then eventually, we started talking about science. And Doug worked on ants and how they form symbioses, these kind of partnerships with other organisms, like plants. And I told him I worked on Streptomyces bacteria. And he told me about these leafcutter ants from South America that grow Streptomyces-like bacteria on their bodies. So this was discovered by a guy called Cameron Curry in the US about 15 years ago. He was the first person to look down a powerful microscope at these leafcutter ants. So it's been known for years they're covered in this white stuff, but nobody really knew what it was. And when you look down a microscope, it turns out that this white stuff are bacteria that look very much like Streptomyces, those long, weird filamentous bacteria. In fact, it's a mixture of Pseudocardia and Streptomyces bacteria. And it turns out these ants are using antibacterials made by these bacteria to protect themselves and their symbiotic fungus against infection, against disease. So Doug and I think Doug had already been awarded a UEA PhD studentship and very kindly agreed to dedicate that studentship to looking at leafcutter ants, and we decided to try and figure out what the bacteria were doing and what antibiotics they might make. And Jörg Barker, who was a German student who came over to do that PhD, really kick-started this whole project. And he was joined by Ryan Seipke, who's an American scientist who joined us as a postdoc and really started up a lot of the chemistry. Okay, so leafcutter ants. So this is one of our captive colonies. And if you visit our website, you can see a live feed webcam. So you can just watch the ants running around, which is kind of fun. So here you can see all these female worker ants. One of them is soaring through a leaf fragment. And what they do in the wild, they dig these deep underground colonies. They keep them at a perfect or constant temperature, so 25 degrees, 80% humidity. They have air vents to the surface to keep the temperature and humidity regulated. And they go out, they live in South and Central America, they go out into the rainforest, climb up into the canopy, cut leaves, bring them back, and take them into their underground nests, and they feed them to this fungus. So leafcutter ants, despite being surrounded by a vegetation, they can't eat plant material, they can't digest it. So instead, between 55 and 60 million years ago, they became the world's oldest known farmers, or they basically evolved agriculture long before humans. So they feed the leaves to this fungus, Leucoagaricus, what we call the food fungus, and the larvae, the ant larvae, graze on this fungus. That's their sole food source. Okay? So they're basically growing their own food just like we are. Because they keep their underground nests at a perfect temperature for growing fungi, other microbes obviously want to get in there and eat this fungus and eat the leaf material. So they have big problems with disease just like we do. And one of the problems they have is this parasite called Escovopsis, and this is also a fungus. And Escovopsis infects their fungus gardens and eats their food fungus, so they compete for food. And the ants constantly crawl through their fungus gardens, this honeycomb structure, sniffing out any foreign fungi. And when they find it, they cut it out of the gardens, they take it away from the nest, they rub their bodies against it, and then they dig it back into the soil. And they rub their bodies against it because they're covered in these antibiotic-producing bacteria that are making antifungal compounds that kill the Escovopsis. So I think it's pretty remarkable. So here we have a nice video of another one of our captive colonies. And here we fed them some rose plants because they love roses, and it makes for really pretty pictures and videos when we feed them roses. So here you can see the major workers that go out foraging. So they cut these leaf fragments. They can carry 50 times their own body weight. They take them back to the underground nests. They feed that plant material to the fungus. And then the minor worker ants cut out the contaminated parts of the fungus garden, take them away from the nests, sterilize them with antibiotics, and dig them back into the soil. So again, it's been known for a while that these leafcutter ants have these openings, these crypts in their exoskeletons, which other ants don't have. So they're covered in these little holes. And it's been proposed that these holes are actually crypts that are used to secrete food for the bacteria. So you kind of wonder, why would bacteria live on the surface of an ant and give it antibiotics to use to defend itself against infection if they're not getting anything out of it? So of course what they're getting is food. And Sarah did a remarkable experiment to see if the bacteria on the surface of the ant really are being fed by the ants. So here we have this cute photo that Sarah took of the ants drinking this glucose solution, so this sugar water, out of a little centrifuge tube cap. So here she's feeding the worker ants 13 carbon labeled glucose. So this is a heavy isotope of carbon. And what she could do was to feed the ants this labeled glucose and then track the 13 carbon from the ants, which incorporate into their bodies. If they're secreting stuff, then the secretions will have 13 carbon in them, and that 13 carbon will be incorporated into the bacteria that live on their surface. And that's exactly what she found. She found that all of the bacteria, the Streptomyces and Pseudonocardia bacteria, and all the other random bacteria that we find on the surface of the ant, they all get labeled with the 13 carbon. So the ants are eating it, it's going into their stomachs, it's incorporated into their bodies, they produce secretions that go to the surface of the ant, and the bacteria feed on those secretions, which I think is pretty cool. So the bacteria are basically getting free food. It's a great place for bacteria to live. There's lots of nice food being produced. More interesting to us, perhaps, are the antibiotics that they make. So this was work that was done by Jörg and Ryan quite a long time ago now. So what they did was to isolate one Pseudocardia strain from a worker ant and one Streptomyces strain from the same worker ant. And here you can see the bacteria are in the surface, in the center of the plate, and the Escovopsis is on the outside. So this is the fungal parasite. And we're really testing to see whether these bacteria can kill Escovopsis, which is the prediction. And you can see that you get a big zone of inhibition, much like Fleming's Petri dish. You get a big zone of inhibition where the fungus can't grow anywhere near the bacteria. And it turns out Pseudocardia make a single antifungal that we call nystatin, P1. So this is a novel antifungal compound that kills Escovopsis. But the Streptomyces make two antifungals, antimycin and candicidin, and probably actually more than that. And this is interesting because what we think is happening is that the ants are using different bacteria to make multiple antibiotics, and the use of multiple antibiotics stops the Escovopsis from becoming resistant to those antibiotics. So this happens in human medicine now. If you use multiple antibiotics at the same time, bacteria can't become resistant, or it slows down the evolution of resistance. So ants have been farming their own food for 50 million years, and we think they've been using antibiotics very sensibly for tens of millions of years as well. They use combination therapy, they vary the antibiotics they use, and the parasite, the Escovopsis, never becomes resistant. So we also wondered why, when you sequence the surface of an ant, given that there are trillions of microbes in a gram of soil, trillions of bacteria, we only really ever see Pseudocardia and Streptomyces bacteria on the surface of an ant. So again, Sarah did a clever experiment where she looked at the genes, the instructions that were being expressed on the by the bacteria living on the surface of the ant. Okay? So she basically wanted to see whether the bacteria living on the ant are making antifungal compounds and antibacterial compounds. And she found actually quite high expression of antibacterial gene clusters, so the instructions to make antibacterials. And it turns out that the Pseudocardia and Streptomyces aren't just there to defend the Escovopsis or to kill the Escovopsis and defend their food fungus. They're also shaping the microbiome, the bacteria that live on the surface of the ant. They're making antibacterials that stop any bad or useless bacteria growing, and they allow only the good bacteria to grow. And here you can see some of these strains, the Pseudocardia strains that we isolate from ants, they're killing other bacteria. So these Pseudocardia and Streptomyces protect the ants against bacterial infection, and they protect their food fungus against fungal infection, which I think is pretty amazing. Okay, so can we find new antibiotics from these systems? And this is work done by Rebecca, who's in the audience, and Chin, who's a chemistry postdoc at the John Innes Center with Barry Wilkinson. And this is the species I talked about earlier on, Streptomyces formicae, this new species. Potentially makes lots of secondary metabolites. And it's a really interesting strain because the things it makes we've never seen before. So when we screen it against drug-resistant bacteria and drug-resistant fungi, we see it has very potent activity. So it's very powerful against MRSA. It's very powerful against Candida albicans, which is a fungus, and also against Lamentaspora prolificans. This is a multi-drug resistant fungus. It causes disease in humans. If you get a systemic infection of Lamentaspora prolificans, you have no chance of survival. There's nothing that can be used to treat it. But this bacterial strain kills it stone dead and also kills MRSA. So just 2 of those gene clusters, so there's 46 gene clusters, and these molecules are encoded by just 2 of those 46 gene clusters, the only 2 things that it makes. Cluster 26 are the instructions to make this antibacterial, which is very novel. Cluster 30 are the instructions to make this antifungal, which we also think is very novel. So we haven't managed to get a chemical structure of this antifungal yet. The antibacterials are really exciting. We call them formicomycins in honor of the ants. And they're, well, I won't go into the chemistry, but basically they're very, very powerful against MRSA. And the really exciting thing for us is that MRSA, when in the lab, doesn't become resistant to the formicomycins. Okay, so we can grow it in the presence of very tiny concentrations of formicomycins for lots of generations, and MRSA never becomes resistant. And this is great because we really want to develop powerful new antibiotics that bacteria can't become resistant to. So then Rebecca did another amazing experiment. So I'm always astounded by the things these guys can do in the lab. So here we used a new tool that's only been around in Streptomyces for 2 or 3 years called CRISPR genome editing. And what Rebecca did, so this shows the wild-type Streptomyces formicae strain. So here you can see the bacteria killing MRSA on a plate. You get a big zone of inhibition. And at the top, a technique called HPLC basically shows these peaks. You can separate out all the chemicals in the cell, and these peaks are the formicomycins. So Rebecca used CRISPR to delete all of the genes, all of the instructions needed to make these formicomycins. So she basically made this mutant strain that can't make formicomycins anymore. The formicomycins disappear, but what happens is the bacterial strain, the Streptomyces strain, just switches on a different antibacterial. Okay? So it can't make formicomycins anymore to kill MRSA, so it just switches on production of another antibiotic that isn't usually made. So this is exciting because we can use this to prevent the rediscovery problem. So we can go in, we can edit genomes now, get rid of the instructions for known antibiotics, get rid of them, and then see what else is switched on. And quite often when we do that, when we stop strains from making their preferred antibiotics, they switch on these cryptic pathways instead and make different antibiotics. So we're really excited about taking this strain apart, and Rebecca has now made a series of big deletions in the genome of this strain to delete all of the biosynthetic gene clusters and figure out exactly what this thing is making. Okay, so I just want to finish by talking a little bit about some work we started about 3 years ago on plants and Streptomyces. So the ants are a really great system for trying to understand how antibiotics are used by eukaryotic hosts in the environment. But leafcutter ants aren't really very useful to people. Plants are really useful to people because they provide all of our food. So this is work started by Sarah, who's now a 4th-year PhD student, and Jake, who's a 3rd-year PhD student, and Sam, who's in just started his 2nd year. So Jake took these amazing pictures. So Streptomyces live in the soil, so they're in every soil on Earth. And the place with the most food in the soil is just around the roots of plants. So plants fix carbon from carbon dioxide, and they secrete about 30% of that carbon through their roots. And it's believed that they do that to recruit beneficial bacteria to their roots so that they can have a healthy collection of bacteria to protect themselves against infection. So we wanted to know whether Streptomyces might be used to protect plants against infection. So Streptomyces make antibiotics. It would be great for plants to have Streptomyces living in their roots because it would obviously offer them benefits like it does with the ants. So the red is a plant root. So this is a plant called Arabidopsis, stained red. And the green are Streptomyces strains that are expressing green fluorescent protein. So again, you can see this filamentous growth that we keep seeing. So Streptomyces, these roots have been really washed very vigorously, and Streptomyces cling to these roots. Okay? They grow very tightly on these roots. Again, if we grow plants, if we grow seedlings on agar plates and we drop Streptomyces onto them, you can see the Streptomyces grow very tightly around the roots. So they love to live on plant roots, okay? Because the plants are producing food. This is a great place for Streptomyces to live. Just like the ants secrete food for their bacteria to live on, the plants are secreting food and the Streptomyces want to eat it. When Sarah does sequencing, so she looked at, so these are 3 different plants, so 3 replicates of the plants. She looked at the soil they're growing in, the rhizosphere, which is the outside surface of the root, and the inside of the root. And red means there's lots of Streptomyces there, and yellow means there isn't very much Streptomyces at all. Basically, we can see there's loads of Streptomyces. They're not just living on the surface of the root, but they're also living inside the plant roots. They're getting inside and presumably doing something useful in there. And this is one of my favorite. Sarah's sent me loads of cool data over the years, over the last 3 years, but I think this is probably one of the most remarkable pictures she's sent me. So here we have sterile wheat seeds, so there's nothing on them at all. And in the center, we have a little plug of agar that contains the Tacol fungus. And the Tacol fungus causes huge problems, huge crop losses in wheat. Okay, so it can wipe out whole wheat fields, which is a problem because wheat is one of our staple crops. So Sarah has isolated lots of Streptomyces bacteria from plant roots, and she screened them all against the Tacol fungus to try and find Streptomyces bacteria that kill this nasty disease. And this strain, so what she did here was to coat the seeds in this Streptomyces strain, and then did exactly the same experiment. Put Tacol fungus in the middle, let the seeds germinate, and you can see that wheat seeds coated in Streptomyces, it's like they've got an invisible force field around them. Okay? The Tacol fungus doesn't grow anywhere near them. And this is really exciting because there's huge interest now from agri-tech companies like Monsanto, Bayer, in using natural biological materials, so natural microbes from the soil, instead of fertilizers and pesticides, to protect crops against disease and to encourage their growth. And we think that Streptomyces are really promising bacteria. So their antibiotics aren't just useful in medicine, but we think they're going to be really useful in agriculture because you can coat wheat seeds in Streptomyces bacteria and protect them against infection. Okay, so I'm pretty much finished. So I hope I've convinced you that actually the future is really bright for antibiotic discovery and searching in new places, combined with all these tools to switch on all those cryptic antibiotics, is going to give us hundreds, if not thousands, of new antibiotics. It's going to take a long time. It's going to take 20 years from now, probably, before we start seeing new antibiotics in the clinic. There's going to need to be major funding from government or pharmaceutical companies. At the moment, nobody wants to pay for this. So keep up the pressure. And also, we are seeing already better antibiotic stewardship. So we have the first generation of antibiotics that we've really misused. A lot of them don't work anymore. So hopefully, when we get the second generation of antibiotics, we'll be a lot more sensible in how we use them. So I want to thank the research councils for funding. So I've been fortunate enough to be continuously funded by research councils since I've been at UEA, and that's all taxpayers' money. So thank you. If you pay tax, and not you students, because I know you don't pay tax. And of course, the people that did the work. So this is my current research group at the top. These are all people that have contributed to the work I've talked about today, and actually a lot of work that I haven't had time to talk about. So we've done loads of other, or they've done loads of other cool things that I haven't even had time to talk about today. I have a whole load of amazing collaborators, and all of these people are on the research part. They're all world leaders in their field, and they're all based here in Norwich, at the university or at the institutes. So I'm very grateful to all of them because we can do interdisciplinary science, so I can work with chemists and plant scientists and ecologists, and we can ask really interesting questions and I think do really interesting work. I particularly want to thank Tracy Palmer. So Tracy Palmer was a Royal Society Fellow at the John Innes Centre when I was doing a postdoc. We got to know each other, and when I started my lab, she really helped me to write my first grant applications. She basically got me my first grant because she just rewrote the grant application for me. She's been fantastic. She's been a great mentor throughout the last 10 years. And actually, she's just been made a Fellow of the Royal Society, which I'm really over the moon about. She's brilliant. Mark Butner, of course, who taught me all about Streptomyces and all of the Streptomyces group at the John Innes Centre. And when I started at the John Innes Centre, the first day I started at the John Innes Centre, Paul Hoskisson started the same day. We worked next to each other on a bench, on a lab bench for 3 years. And Paul Hoskisson went off to Scotland and started his own group, and he's just been made a professor as well. So I'm really chuffed about that. And this is a picture of the master, which is Mark, and his apprentices. And we look really happy in this photo because we just left the Fat Cat. We just had 4 hours worth of beer in the Fat Cat. So we're pretty happy at that point. And of course, my family. So my mum has always been a great support to me. And these little chaps who are in the audience, go on, stand up. You want to stand up? No? Oh, we've embarrassed them. All right. Well, they're amazing anyway. So Noah is 9 now and Elliot is 6. They're football mad and they're both brilliant footballers. So anyone that knew me when I was at the John Innes Centre would know I had no interest in football whatsoever, but now I'm completely addicted. We do football training Thursday, Friday, Saturday, Sunday, and football matches in between. We have Norwich season tickets. Even Kerrion has to go and watch Norwich City now. Which just lets me finish on the fact that Norwich City are top of the league, which makes me happy. Thank you very much.
Good evening, everybody. So I'm just here to wrap up the evening. So I'm Andrew Burke. I'm Professor of Evolutionary Biology in the School of Biological Sciences and Director of Research in the school. So I'm going to first take some questions for Matt, then I'll give a brief vote of thanks, and then I'll invite you all to join us in the reception in the lobby outside. So for the questions, let me tell you, for the purposes of the mic, I will have to repeat the question, okay? It's not that you're necessarily unclear. So let's have some questions, please.
Matt, first of all, thank you for a great talk. In the case of formicomycin and MRSA, do you know the cellular target for formicomycin and why it's not possible for MRSA to develop antibiotic resistance?
That's a very good question.
Let me
sorry, you
repeat
want to repeat it?
Do you know the cellular target in MRSA?
Yes, so that's a really good question. I didn't really talk about it today because there's some IP issues, I guess. None of it is published yet. So we think the formicomycins are targeting two different processes in the same cell. So all the antibiotics we know about only inhibit one essential target in a bacterial cell. We think the formicomycins inhibit two different essential processes at the same time. And actually, I forgot to say, the BBSRC just awarded us a grant, just over £1 million, to try and develop those antibiotics further. So Rebecca will be carrying on after her PhD and joining us again in July to work on that project.
The questions, please.
Hang on, you're one of my PhD students. This looks like a plant now. Right?
No, I was going to say, ants, plants, where do you think is next? Where's your next big exciting environment to try?
I think the plants are going to keep us busy for a long time. So I naively thought it would be interesting to start working on plants without realizing quite how complicated the whole thing is. And I don't know anything about plants, so I think...
Try the inside of football boots.
Yeah, that would be good. I think plants will keep me going until I retire, I think, if I get to retire.
I'll have to repeat. I forgot to repeat the last one. Forgive me. Are there any other? This one is. Are there any other insects similar to leafcutter ants? I guess in terms of their relationship with bacteria and fungi.
So there's a few examples, but I suspect it's probably widespread and people just haven't looked hard enough. So there's some amazing insects called digger wasps that actually have. So they keep streptomyces in their antennal glands, and when they lay their eggs and their larvae hatch, they spray streptomyces all over them, and they incorporate the streptomyces when they spin their cocoons, and those streptomyces produce antibiotics to protect the developing larvae from infection, so that when they emerge as adults, they're healthy. If you get rid of the streptomyces in that system, they never emerge as adults, they just die from fungal and bacterial infections. So that's a really well characterized and interesting system. But I think there's probably lots more, and people haven't looked hard enough. It's a good question, thank you.
One over there.
What do you think about using bacteriophages to cure diseases instead of antibiotics?
So what do you think about using bacteriophages to kill diseases instead of antibiotics?
So bacteriophage were actually used before antibiotics were introduced. So certainly in Russia, they were quite widely used. The problem with bacteriophage is they're very narrow spectrum. So they only target, so a particular bacteriophage will only target a particular strain or species of bacteria. And traditionally, people have tried to come up with broad spectrum antibiotics that would kill all bacteria. So if you knew what the infection was, if you knew what the disease was caused by, and you knew exactly what kind of bacteria it was, then phage might work. The other issue, of course, is convincing people to swallow a virus to kill a bacteria, which might be an issue. But there have been some famous cases of people. So there was one in the US recently where somebody saved her husband's life by applying a cocktail of bacteriophage. So I think it could work. What do you think, Laura?
I think there's potential, and I wouldn't rule it out, and I think we should explore it.
Definitely.
Any further?
You said you were against using antibiotics to promote growth in cattle, and then do you think it's
Okay, the question is, if it's wrong to use antibiotics to promote growth in cattle, why is it right to use them on wheat plants?
Okay. So I guess there are two issues here. So actually, some Streptomyces natural products are used in purified form and sprayed onto crops and used as pesticides or fungicides. But what we're talking about is using the natural microbes and encouraging them to grow with the plants. So you wouldn't be using huge amounts of purified antibiotic. You'd be letting the bacteria do the work. So it's a more natural system, I would say. Thanks.
Okay, perhaps one more question?
One at the front there.
Should we be doing anything to control overpopulation before we introduce antibiotics that could potentially save lives?
Well, that's the elephant in the room, isn't it?
Should we be doing anything to control overpopulation before using antibiotics to cure diseases further?
Yes, we should, but it's something that nobody wants to talk about, controlling population, because then you get into dangerous areas like eugenics and, do you have the right to stop people having children? All those kind of questions. So some people would argue that disease is a natural population check. I'm not getting involved in that debate.
Okay, we'll leave the questions there. Thank you for them. So I'd just like to finish up then with a vote of thanks to Matt. And it's really a great pleasure on behalf of the school and I guess the university as well to thank Matt for his inaugural lecture, which I think, well, you'll agree was both fascinating and stimulating. And we also heard about the excellent and innovative research that Matt and his research group and collaborators have been doing. But I'd like to take a few moments to tell you a few things you might not have known about Matt's achievements and qualities.
Okay, I'm worried now.
So I'm just going to focus on two of these. So first, not everyone might know that Matt was a drummer in a band called Bear Suit. And you can find out all about it, and I guess listen to some of it, on Wikipedia, in fact. But maybe, his willingness to take to a stage, I think is reflected in something we haven't heard so much about, but we've seen tonight, which is his very great passion for science communication. So he has been a tireless ambassador for this, and specifically, of course, for research on antibiotics, taking his message to mass audiences through media interviews, his lab's AntCam, which we heard about, and stands at such prestigious events as the Big Bang Science Fair and the Royal Society's Summer Science Exhibition. And he has a very great, not just tireless energy, but a very great talent for science communication. And he's even sometimes combined his love of science communication and his love of music by going to communicate science at music festivals. At least that's what the expenses claim say.
Yeah, that was a good one.
So second, running a successful and harmonious research group, and we felt that listening to this talk this evening, relies on many kinds of unsung skills in academics, I would say. And members of Matt's group do report the enduring concern he shows for both their academic and their personal welfare. So on the academic side, as we've seen, he has a very broad interest in his field and is a fount of new ideas reflected perhaps in the variety of taxa, different kinds of organisms that he studies in his lab. Plants, insects, bacteria, fungi, and even those relatives of the bacteria, the archaea. And on the personal side, Matt looks out for his students and other group members. It's been reported the way he might for his family. And to match the interests of those around him, as we've heard, he even has taken up an interest in football that he told us about, and even to the extent of rooting for Norwich City Football Club. And Personally, I would never go there. But there we are.
Actually, come, it's fun.
So I'd just like to say, Matt, many congratulations on your professorship, and please could everyone join me again in thanking you for your inaugural lecture. And now you're very welcome to join Matt and everyone else for the drinks reception in the lobby outside.
Thank you. Thanks, Andrew.
You're welcome.
Drink time.